This is the Tricorder challenge for biochemical sensors. My name is Chad Austin, and this is the overview slide for the Tricorder. And on the left, you can see three biosensors for different disease conditions. And on the right, we have a sensor for pandemic infectious agents, and then we have a sensor for chemical agents. So the first biosensor is a breathalyzer for COVID-19 detection. The second biosensor is a surface plasma and resonance-based alpha-synuclein sensor for the early detection of Parkinson's disease. And the third biosensor is a surface-enhanced Raman scattering multi-cancer urine sensor. For the pandemic infectious disease sensor, I've selected an avian influenza capacitive biosensor. This is the H5N1 type of flu, also known as bird flu. And then the fifth sensor in the tricorder is for chemical agents, and I've selected a FET-based synthetic opioid detector. Specifically, it's for fentanyl. Fentanyl has become an occupational risk for first responders, and it's also critically important for first responders to assess substances in a situation to detect what a patient might be suffering from if they're unresponsive or unconscious. So the first biosensor is the breathalyzer for COVID-19 detection. So this is an electrochemical sensor. As mentioned, it detects COVID-19, but it's also capable of being modified to detect other coronavirus infections. The motivation was the lack of a breath-based sensor for COVID-19 and other respiratory diseases. Many of them are swab-based or require liquid. So the target analyte is the SARS-CoV-2, the binding protein, the receptor binding protein, spike protein. And so that is binds to the receptor binding domain of a target cell. So that is what's being measured. And SARS-CoV-2 is just the name of the virus that caused the COVID-19 pandemic. The medium is the breath, but it does get aerosolized in the sensor. And the analyte concentration, at least in the research, was from one RNA copy of the virus all the way up to 10 to the 6 copies per microliter. So the sensor performance during the lab testing had a specificity of 100%. It was quite high with a sensitivity of 98.4%. The core of the technology is the molecular imprinted polymers. So the molecular imprinted polymer layer with a selective recognition site that binds with the virin receptor binding domain spike proteins. The binding of the virus to the cavities reduces charge carrier mobility in the surface of the layer on top of the electrode, and then that will increase the ohmic resistance, which can be measured. The detection limits are from 0.1 to 1 femtogram per milliliter. So it's quite small, and this translates to about a 0.1 to 1 RNA copy of the virus per microliter. So the main benefits are super fast. It takes about 30 seconds. It's breath-based and has low power, and it's cheap and it's portable. It's made from cheap components. Some of the main limitations are the environment can affect at precision. So the environment could have some effects on those binding domains on the MIP, and also mutations in the virus could reduce binding affinity. So as a wild type virus changes in that conformation of the receptor binding domain spike proteins changes, this won't be as selective. So it's analogous to an artificial antibody, these cavities. And so in the upper right hand, you can see the fabrication flow process here. Um, so initially there's a substrate, like a glass or silicon dioxide substrate, and then there's a bottom electrode that's laid down through electrode deposition. Um, and then there is the graphene Prussian blue layer that then is put on top of the first electrode. And then there in a PBS solution, they combined basically it's the template molecule, and that is the spike protein you see on the left part. And then that's combined with dopamine. So dopamine provides a functional monomer, which helps with some of the binding properties in that cavity. So it's mainly a type of hydrogen bonding. Um, so that it in that S protein templates will help match the shape and size with a strong hydrogen bonding between the cavities and the analyte molecule, which in this case is the spike protein, the virus. So the polypyrrole then is formed through an electrochemical deposition process around the spike protein. So that is laid down and is sort of molded around. And then once the template is removed, you have these bind domains there that are analogous to an artificial antibody. So that's in the middle bottom picture. And then the last the last picture, number 5, you can see in the lower left, it shows the COVID-19 virus that is bound to these cavities. And when that happens, as mentioned, that's going to reduce the charge carrier mobility, and that the resistance will change in the electrodes, actually going to increase. And that could be detected just through, common multimeter, whatever measuring method can be used in the device. Um, that is the link to the article, and the commercialization is currently in a research phase. Although some of the authors are affiliated with Winchester Technologies that do microfab and some RF miniaturization, but there currently is no commercial device employing this technology. Some of the competing technologies are reverse transcription, PCR, although this can take a lot longer. And this is also more expensive. And then Inspect IR has a mass spectrometry based sensor. So it's a combination of mass spectrometry with machine learning based um, techniques. So that is the main competitor at the moment. The second biosensor is a surface plasma and resonance based alpha-synuclein sensor. So this is for the detection of Parkinson's disease. So alpha-synuclein is a major biomarker of Parkinson's disease. In fact, aggregations of alpha-synuclein are responsible for the areas of the brain and central nervous system that get destroyed during Parkinson's disease. And there's a major prodromal period for Parkinson's disease where alpha-synuclein can be detected and affecting the body, 10 years before Parkinson's manifests. And so early detection is quite important. So the alpha-synuclein protein is the main analyte in this sensor, and the medium's blood serum. So it's more convenient in that it's not super invasive, it just requires a blood draw. So the analyte concentration tested in the experiments was 3.6 plus or minus 2.53 picograms per milliliter in plasma, and 0.031 plus or minus 0.04 picograms per milliliter in the serum. So the sensor specificity in this paper was listed in recovery rates. So at a 94.5 to 104.3 recovery rate of alpha-synuclein in the serum. So the 104.3 just kind of indicates that there was some noise. It over detected the the analyte. So this isn't a specificity number. It's just the recovery rate. And so, but that indicates a quite good sensitivity. So it works through surface plasmon resonance, and it which can detect the binding of alpha-synuclein to rabbit monoclonal antibodies. So the sensor is essentially a gold film, and label-free iron oxide is immobilized on this gold film. So there's nanoparticles of the iron oxide, and the iron oxide increases the sensitivity. So metals bound to the gold foil oftentimes will increase the local refractive index of the gold surface, and then that leads to greater sensitivity. And it's mainly because the iron oxide has a high refractive index. Also, iron oxide has a low optical loss. And the rabbit monoclonal antibodies were chosen over murine monoclonal antibodies because in experimentation they were increased the sensitivity quite a bit. So and they're also more selective. They're much more selective to the alpha-synuclein protein than the antibodies derived from mouse models. So the benefits of the detection limits are 5.6 femtograms per milliliter is the detection limit, and that is quite low. So it is very, very sensitive. The main benefits are that it is ultra sensitive, it's real time and rapid and reusable, and it has um, limitations are terms that it requires diluted serum. So yeah, the serum needs to be diluted about 1 to 1,000. So it's quite diluted, and that's somewhat of an inconvenience. It also doesn't have clinical validation yet. Um, in the upper right is a diagram of the sensor. So um, the configuration is on the very right there. You can see in the OLED is the light source for the SPR, and that is transmitted through a prism, and then there's a reflection and refraction, and there's a collimator connected to fiber optic that then detects the signal from the film. And there's an input, there are flow cells on top. So that would have the blood serum and it passes over the reaction chamber. So the inside of the reaction chamber is shown there on the left. So the reaction chamber, you can see there's a substrate of the prism there with chrome film, and then there's the gold film on top. So there are self-assembling monolayers on top, and then in the fabrication process, and then the the iron oxide nanoparticles are those little black circles you see at the very top sticking up. And so immobilized on those are a set of antibodies, and then there is the alpha-synuclein protein, and then a second set of antibodies is provided. So that just increases the sensitivity. So the way this would work is that you would have antibodies already fixed on the iron oxide nanoparticle, and then you would apply your sample, and then the sample containing alpha-synuclein. And after your sample has been pass through the flow chamber, then you would have an additional fluid put through with other antibodies. So then those antibodies would attach to the opposing end of the protein. So the supporting link is shown below there, and the commercialization it's currently in the research stage. So the main competing technology is the ELISA. But as you can see, the ELISA only has a limit of detection of 30 to 300 picograms per milliliter, which is small, but it's an order of magnitude or 3 orders of magnitude larger than the limit of detection for this sensor. ELISA is also a lot slower. It takes hours, where this can be done in minutes. And it also requires labeling, and this is a label free. There's no fluorophores on the required. Other competitors are electrochemical biosensors, Raman spectroscopy, and fluorescent labeling. The third biosensor is a surface-enhanced Raman scattering multi-cancer urine sensor. So this sensor is capable of detecting several forms of cancer. So pancreatic cancer, prostate, lung, and colorectal cancers can be detected by this sensor. The target analyte includes urinary metabolites, and which are there's quite a variety of them that can be measured. So that include amino acids and their derivatives, such as creatinine, tryptophan, tyrosine, phenylalanine, urea. Could be organic acids such as uric acid, hippuric acid, and various nucleosides and nucleotides. Other small molecules such as lactate and acetate associated with cancer can be also detected. So this sensor is convenient because it requires just whole urine that doesn't need to be diluted or treated or modified in any way. You can just work off a urine sample. So the analyte concentrations in the research range from 1 to 1,000 nanomoles in the urine. And the overall specificity of the sensor was 95.6. So the specificity and selectivity was also dependent on the specific cancers. They each had their own specificity and selectivity. So the technology works through, as indicated, surface enhanced Raman scattering. So the metabolites in the urine diffuse through a unique porous 3D evolutionary gold nano architecture. And the structure is placed at the bottom of receptacles. And a Raman laser excites the plasmonic hotspots in this 3D structure. So the surface enhanced Raman scattering is then used to obtain a metabolic spectral fingerprint. So the metabolites, as they've diffused, there'll be electromagnetic interactions at these nanostructures. And then a convolutional neural network is then used for cancer classification. So a binary discrimination algorithm such as linear regression is used to separate non-cancer from cancer categories of urine, and then a convolutional neural net is then used for um, classifying specific types of cancer. So the detection limits are about 1.23 nanomoles of the urinary metabolites, and the main benefits is multi-cancer. So this sensor is capable of, indicating the four types mentioned above. It's non-invasive and it's label-free. The main limitation though is requires a Raman spectrometer, which is quite expensive. It also requires convolutional neural net training, so this can be time consuming. And another possible limitation is that it can only detect the metabolites of a cancer, but it cannot indicate the stage of the disease, give any indication of that. So that is sort of a limit. In the upper right, we can see the 3D evolutionary gold nano architecture platform. And this is the sort of processing flow. So the urine sample is collected, and then it's put into a structure of 96 well plate platform. The nanostructure consists of there's three kind of components to its fabrication. So there's the initial um, gold nano sponge, and then gold nanoparticles are sort of deposited on that. And then after the gold nanoparticles are placed within the sponge, there's sort of a third kind of gold laminate that's put on. And so this creates a lot of granularity in the structure, and that results in a highly volumetric electromagnetic field. So there's all these tiny structures within the sponge, um, and these hotspot sites interact with the metabolites that diffuse throughout the structure. Uh, so the throughput from that, then you can see there's the Raman shifts depending on the type of metabolites. And then this information is fed into the convolutional neural network that then is able to classify the sample into a particular cancer based on the signatures and the from the Raman scattering. So below is the supporting references. So there's the publisher's link. Uh, like the other sensors, this is also currently it's in a research stage. The main computing technologies are liquid chromatography, mass spectrometry, um, and the ELISA and the MRI. But as mentioned, the mass spectrometry, the ELISA can be time consuming and require expensive equipment. And the MRI imaging also is somewhat time consuming and doesn't have the always have the best resolution, and also requires large and expensive equipment. So it's not feasible for a tricorder design such as this. This slide describes the pandemic infectious disease sensor. And as mentioned in the beginning, in the overview, the avian influenza capacitive biosensor is what is used in this. So again, the disease is avian influenza, also known as bird flu, and this is the H5N1 variant. The main advantages of this, what the concern is, there are two main reasons. One, we continue to have outbreaks of bird flu that are costly for poultry, livestock producers. Routinely they lose animals where there's big outbreaks of this, and it also is spread to other livestock in recent years. And then the second, the more important reason is that the pathogenicity of this virus is pretty severe when there has been small outbreaks. There hasn't been much human to human transmission of this virus. It's mainly people who have worked closely with livestock who've gotten sick, but it tends to be much more virulent than other types of flu and certainly than COVID-19 was. Uh, the main target analyte is the H5N1 virus, but specifically is the hemagglutinin surface protein. So that is the H5 part in the H5N1. And the medium in the study, it was an aerosol. So this is able to take in ambient samples. So this is sort of an ambient sensor for, influenza outbreak. This is not for diagnosing a patient. However, in the study, they used aerosolized PBS. That was in the testing of this, the inactive virus they used. They had put in a solution of PBS and then aerosolized it to kind of simulate airborne. The analyte concentration was 2 to 1.6 times 5 to the 10 to the fifth virus RNA copies per milliliter. And the sensor specificity, it didn't list a quantitative number in this research, although it was very selective for the H5N1, and that was because they used H5N1 and aptamers in this. But those antibodies and aptamers are really, really selective for a given genetic sequence or protein conformation. So I think in the paper they've compared this against some other viruses, and that showed no detection where this one did. So the technology is mentioned, it is a capacitive technology. Um, so there's a lattice of Prussian blue and graphene oxide, and that is grown on a silk screen printed carbon electrode. And that the Prussian blue has a high capacitance, but doesn't form lattice structure very well on its own. So it's sort of interspersed within the graphene oxide. And the whole structure has a known capacitance. And then when the H5N1 antibodies are mobilized on the surface of this lattice. So when the virus and the sample is provided and that virus interacts with the antibody, that capacitance will decrease when the virus binds to the antibody or the aptamer. The capacitance can be measured through the electrode with just standard, electronics, and then therefore detection can be obtained through that method. So it's an electrochemical capacitive biosensor. The benefits, the detection limit, so it was able to detect 56 copies per milliliter, or in a volumetric sense, it was 93 copies per cubic meter of air. So the sensing of this was pretty fast. It was less than 5 minutes. They also did an E. coli version of the sensor, which is also less than 5 minutes. It's label free. There's no fluorophores involved, and it's versatile, simple, and it's easy to use. It's not complicated. You can package this into a small electronics, uh, so it fits into the tricorder very well. The main limitations require some kind of nebulizer currently and a single use. Each kind of sensor component once the virus is bound to the antibody. So the upper right shows a schematic of the capacitive biosensor platform. And so the working electrode of the screen printed carbon electrode is shown there. It's the big gray, flat box structure. And then the graphene oxide Prussian blue lattice is shown at the very bottom. And then from that, there is immobilize the antibody, and then that antibody shows it interacting with the H5 protein then of the influenza virus. So, and then that's just sort of a capacitance diagram. So you have capacitors in series, and then there's a very simple equation to kind of relate that out. Um, they're to get the detection information. So the currently it is not commercialized. It's in the research stage. And the main competing technologies are PCR based. There's a company, I think Fisher Science has the aerosol sense. And then there are a few other ones that are similar, but they are more expensive. And again, the PCR is not as fast. It's not less than 5 minutes. The Inspect Aura breath sensor they are trying to port technology towards this type of thing. We had already seen they also have a COVID-19 breath sensor. We saw that in the other slide. And then there's other, electrochemical sensing methods, in general, or competing technology. The fifth and final sensor, the tricorder, is for a chemical agent, specifically fentanyl, and this is the FET-based synthetic opioid detector. So it detects a threat of toxic exposure to fentanyl. So this is a concern to first responders and police. There have been cases, police on, in either apprehending a suspect or at a crime scene are exposed to powder, which could contain fentanyl, which is a synthetic, cheap and highly potent opioid, and they've gotten sick, and sometimes it can be fatal to touch it. Also, with first responders, paramedics and EMTs, they might not know exactly what somebody has what they're suffering from and if they're unconscious. And so if there's a substance there, they're able to test it, and they can detect it's fentanyl, something that might, increase the time and how to respond. So the target analyte was the fentanyl, the molecule itself. The medium, it needed to be put in a PBS buffer solution. So it's not able to detect in a solid form or dry form. And the analyte concentration, the testing was 2 to 50 micrograms per milliliter without the antibody. And they did testing of 10 femtograms per liter to 100 picograms per milliliter with the antibody. So as you'll see, there are two main ways they did this test. So the sensor specificity was 91.2% versus other opioids when combined with a machine learning classification algorithm, and they use linear discriminant analysis. So the technology, the basis of it is this field effect transistor. So there are single-walled carbon nanotube network that are put between the source and drain electrodes of the FET. And when the fentanyl binds to gold nanoparticles that are embedded within the carbon nanotubes, um, this causes end doping and a decrease in conductance. So this can then be measured electronically when the conductance changes. So the sensitivity and selectivity are both increased and amplified when fentanyl antibodies are immobilized on those gold nanoparticles. So the detection limits are 10.8 femtograms per milliliter and 32.1 femtomoles. And it's ultra sensitive and highly selective and very fast. The main limitations are that it's only been tested so far to using, in a PBS solution in the lab. The upper right shows a schematic of the single walled carbon nanotube FET. And you can see there is the silicon and silicon dioxide substrate. And on both ends of the substrate, you can see the source and the drain electrodes. And then between them is this network of the carbon nanotubes. And then embedded on them are the gold nanoparticles. Uh, when the antibodies were mobilized, they would be on those gold nanoparticles. It is not shown in this diagram. And then to the right is just an atomic force microscopy image of the carbon nanotubes without the gold nanoparticles in the very top. And then there's an image and the gold nanoparticles are embedded within the carbon nanotube structures. The device is currently just in a research stage. The main competitors are gas chromatography, mass spectrometry, electrochemical fentanyl sensors, and surface enhanced Raman scattering. But yeah, this device has better quantification than the surface enhanced Raman scattering, and is more sensitive than most of the other chemical sensors currently available. And that concludes the presentation of my tricorder. Thank you for your time and thanks for listening.